Welcome to the lecture on DNA, the master molecule of life. DNA is often called the blueprint of life. Take a moment and think about why we would call this molecule a blueprint. A blueprint contains instructions for a building, or in this case, the Batmobile. And our DNA contains instructions for building an organism. It tells us how to build cells, where to put them, and what to do with them once they're made. So these are just like building instructions for this Batmobile. It shows all the needed parts and how they fit together. The DNA in the nucleus gives instructions on how to build proteins. And proteins are how our traits, such as eye color and skin color and hair color, are all expressed. Our proteins also do a lot of functions in our bodies to make sure that our food is being digested and that our bodies can move. It's what our tissues are made of. Proteins are very, very important for our bodies, and DNA is the instructions on how to make all of them. So why do we study DNA? Please pause the video and come up with 2 to 3 reasons why we would want to study DNA. If you're having trouble thinking of them, or would like to check and see if your answers are accurate, please contact your instructor. We study DNA for many reasons. For one, it's of central importance to life on Earth. That is one of the key things that makes organisms alive, having DNA. Also, there's lots of medical benefits, such as cures for diseases and making better food, better yield on crops. Being able to manipulate DNA allows us to manipulate life, and it makes working with organisms and getting the best output possible much easier. Chromosomes are composed of compressed DNA, and that's DNA is broken into segments called genes. So a gene is a series of nucleotides, or just a small segment of DNA. As is shown on this picture, there's only a few nucleotides that would code for hair color. Or here's the gene that tells our organisms how to have teeth developed. Or a gene for a specific protein, maybe that's used for digestion, or for eyesight, or for some other transport within your body. The typical human cell contains about 80,000 genes to tell those cells how to work on their own, but also how to work together with other genes. Each human cell contains 23 homologous chromosomes, or 46 total. So those 23 homologous chromosomes are paired up with 23 other chromosomes that are the same chromosome, but with possibly different genes on it, or different alleles in the same gene. So we have 22 pairs of autosomes. Autosomes are cells that are not sex chromosomes, and 1 pair of sex chromosomes, either X if you're female, or XY if you are male. We can also group cells into 2 groups called somatic cells, which would be diploid and haploid. Somatic cells are diploid. Diploid means that there are 2 copies of each chromosome within that cell. That's how we get 46 chromosomes, is those 23 are paired up with another 23. Haploid cells have only 1 copy of those chromosomes, and therefore we call them haploid. Make sure that you can use those terms to describe cells. Look at the picture and see if you can answer the following questions. Where is that DNA coming from? What is the structure that it's leaving? In what other organelles pictured in that picture do you think have DNA in them as well? How would the DNA condense into a chromosome? And how many of these are in each human cell? The DNA is found in the nucleus. That's what this is coming out of here, is showing the DNA leaving the nucleus. Some other organelles that have DNA are actually mitochondria, which are these purple ones shown here. And chloroplasts also have their own DNA. Since those are only found in plant cells, they are not pictured in this picture. DNA condenses into chromosomes by wrapping around proteins called histones, and we'll get to that in just a moment. And how many of these are in each human cell? There's 46 in each cell, 23 pairs of each 23 chromosomes, and there's a pair of each, so 46. The shape of a DNA molecule is a double helix. What's a double helix? Well, it's kind of a long, twisty, rope laddery type thing. If you were to unwind it, it's like a twisted ladder, okay? Or maybe a zipper. DNA is called a polymer because it is made up of many pieces. Poly means many, and it's made up of monomers called nucleotides. The type of organic compound this is a nucleic acid. DNA and RNA are the only two types of nucleic acids that there are. A double helix shows two strands twisted together. So the nucleotides on each strand are held together with covalent bonds. That's what these lines, the solid lines here, represent, are covalent bonds. There's covalent bonds between the sugars and the phosphates, and there's covalent bonds between the sugars and the bases here. Between them, here are hydrogen bonds. These are held together here in the middle with very weak bonds. The hydrogen bonds are easily broken, and that's a good thing so that the DNA can unwind to replicate or to make proteins. We can get the code from it. Complementary bases are bases that are pairs. Think of things that you know of that are complementary. The first thing that comes to mind for me is peanut butter and jelly. Those are two things that just really belong together. They're not the same, but they fit well. Try and think of something else that is complementary so that you can remember this later. Here is what DNA kind of looks like. These yellow circles represent phosphates. These blue shapes here represent the sugar, and these shapes here represent the bases. One of these three things together comprise a nucleotide. So DNA, the backbone, this part here, is alternating phosphates and sugars. Phosphate, sugar, phosphate, sugar, phosphate, sugar. The base is only attached to the sugar. The base is not attached to the phosphate. So we have this alternating phosphate here in the middle, but there's kind of a space between the bases. There's no base attached to any of the phosphates. DNA shape, so then there would be another side mirroring this with a complementary base, a sugar and phosphate, and then these two would be twisted together and wrapped around histones. Histones are proteins that help the DNA to prevent from tangling, and those subunits, the DNA with the histone together, is called a nucleosome. A nucleotide is the nitrogen base, the deoxyribose, and the phosphate. This is the monomer of the DNA or RNA. These three things together compose a nucleotide. There are 4 different types of bases, and so 4 different types of nucleotides. There could be adenine, thymine, guanine, or cytosine. So therefore, depending on the type of base that is attached to the sugar, that is the type of nucleotide it is. These 4 different bases are often just represented by their letters. Cytosine and guanine always pair up, C and G. It helps me to remember that because the letters kind of look the same, so they pair up. Thymine and adenine pair up. Chargaff's rule, Chargaff was the one who figured out that adenine and thymine always pair up and cytosine and guanine always pair up. So his rule is that there's always equal numbers of adenine and thymine, and there's always equal numbers of cytosine and guanine in any given DNA strand. That's because since they're always paired up, they always have to have equal numbers. The adenine, however, does not have to equal the same amount of cytosine. So adenine and thymine are always the same, but it doesn't have to be equal to the cytosine and the guanine as well. Here's just some fun little facts about DNA. The average human has about 60 to 90 trillion cells. So that's a lot of cells that make up your body. Think about a trillion. That number is kind of hard to comprehend. It's not something that you can really manipulate or that you ever really work with. So just to give you an idea of how large a number a trillion actually is, if you started spending $1 million every day since Jesus was born, you still would not have spent $1 trillion. So that means you've lived for 2,000 years and you've been spending $1 million every day. You still have not spent $1 trillion. That's a lot. Trillion is a huge number. So each cell contains about 2 meters of DNA. And in your 60 to 90 trillion cells, that means that your DNA can travel from the Earth to the Sun 400 times. That's a lot. That's a really long way to travel. But the diameter of your DNA is super, super, super small. 0.000000002 meters. Very, very small. To conclude, you do need to know some of the scientists and their basic ideas. We talked about Chargaff a little bit in this program here. Watson and Crick are the ones that discovered the structure of the DNA. Please use chapter 8.1 to read about Griffith, Avery, and Hershey and Chase and Franklin and their experiments, because they're also important and to know what they did, but we're not covering them in the lecture. You need to do that from your reading. That concludes our lecture for today, so if you have any questions, please contact your instructor, scroll down, and complete the assignments for this week.